Hi guys, I'm Edmund. So today I'm gonna talk about the rotavirus with my group leader Clement. Rotaviruses are the most common cause of diarrhea disease among infants and young children. Nearly every child in the world is infected with a rotavirus at least once by the age of 5. Okay, so now we're gonna talk about the structure of rotavirus. The image A is the electron micrograph of rotavirus triple layer particles, and the image B is the cross-sectional schematic of the rotavirus triple layer particles. The rotavirus structure consists of the inner capsid layer, middle capsid layer, and the outer capsid layer. And from this image, we can see that the rotavirus is mainly made up of viral protein, and each viral protein have different function. Like viral protein 7 and 4, they are responsible for the production of the rotavirus specific antibodies. And the image C is the electrophoretic migration profile of the rotavirus double-stranded and the encoded protein NSP, which is non-structured protein. Okay, so the next part we're going to talk about is the replication cycle of rotavirus. At first, the rotavirus is attached to the host cell surface, followed by the interaction with other cellular receptor. The rotavirus is then entered to the inside of the cell by receptor-mediated endocytosis. The low calcium of endosome triggered the uncoating of the rotavirus. The uncoating process results in the release of transcriptionally double-layer particles. The double-layer particles start to synthesize mRNA transcription, which is used to translate viral protein. Once the amount of viral protein reaches a specific amount, the RNA genome is replicated and packaged into newly made double-layer particles in specialized structure called viroplasms. The newly made double-layer particles is then bind to NSP4, which serve as an endoplasmic reticulum receptor. The transition membranes are removed as the outer capsid proteins, viral protein 4 and 7 assemble, resulting in the maturation of the triple-layer particles. The replicated rotavirus is then released through the cell lysis. Okay, so the last part of my presentation is about the rotavirus symptoms. A rotavirus infection usually starts within 2 days of exposure to the virus. Initial symptoms are fever and vomiting, followed by 3 to 8 days of watery diarrhea. The infection can cause abdominal pain as well. In adults who are in perfect health condition, a rotavirus infection may cause only mild signs and symptoms or none at all. There are some common symptoms between the child and adult, which is diarrhea for more than 24 hours, high body temperature, and frequent vomiting. Okay, so now this is the end of my presentation. The next part of the presentation, I'm going to pass it to my group member, Clement. Clement, please.
I will now be presenting on the transmission of rotavirus, which is mainly through the fecal-oral route. The transmission of rotavirus starts with defecation, where stools shed during rotavirus-associated diarrhea contain large quantities of the virus. Any surface that comes into contact with these stools, including your hands or the toilet seat, becomes contaminated, and this contamination can spread from surface to surface until finally a contaminated surface reaches a person's mouth and the person becomes infected. Transmission of rotavirus through airborne droplets has been speculated but have yet to be proven. Now, how is rotavirus infections diagnosed? Rotavirus infections are hard to diagnose accurately as they are clinically indistinguishable from other diarrheal diseases. However, there are certain factors that can point to rotavirus infections, such as the severity of the infection, as rotavirus infections are generally more severe than other diarrheal diseases. The seasonality of the infection is also very important, as certain pathogens may only be active during certain seasons. For example, if a child has diarrhea during winter, rotavirus may be the cause of it. For laboratory confirmation, rotavirus antigen is detected in stools through electron microscopy, polyacrylamic gel electrophoresis, and antigen detection assays. How is rotavirus treated? Treatment is only required in severe cases with intense vomiting, repeated watery diarrhea, and high fever. And treatment is done through fluid and electrolyte treatment, where oral rehydration salt containing glucose and several salts is fed to the patient to treat dehydration. Probiotics are also used, including lactic acid producing bacteria and yeast. Dietary management is also very important, as adequate protein calorie intake must be maintained, as a lot of nutrition is lost during diarrhea episodes. Zinc supplementation and lactose-free milk formulas are also encouraged. Antiviral and antiemetic drugs are currently being studied as therapies to treat rotavirus infections. The only way to prevent rotavirus infections are through vaccination, and rotavirus vaccines are live vaccines which causes the body's defense system to produce antibodies. Vaccines have been proven to be very useful as they prevent 74% to 87% of all rotavirus diseases. Common side effects include diarrhea, vomiting, and increased irritability. The history of rotavirus is very short, with its first observation in 1973, its naming in 1974, the first vaccine being licensed in 1998, and the second vaccine being licensed in 2006. These are the references used during our research. Thank you for your time.